Welcome to the Wheats on Your Mind podcast. My name is Aaron Harries. Wheats on Your Mind is brought to you by the Kansas Wheat Commission and Kansas Association of Wheat Growers. On this episode of the podcast, we are visiting with Bikram Gill and John Raupp with the Wheat Genetics Resource Center at Kansas State University. Bikram Gill was born and raised in the state of Punjab in northern India. He received his PhD degree from the University of California, Davis. He joined the faculty at K-State in 1979, and he earned University Distinguished Professor in 1997. John Raupp has managed the Wheat Genetics Resource Center and wild germplasm for the Wheat Genetics Resource Center Gene Bank since August of 1980. He's developed it from a small collection of 300 lines to over 10,000 as present. And we're glad to have John and Bikram join us on the podcast today. This is something I've been wanting to do for a long time. We're going to talk about how it all began, the origins of bread wheat. So it's a very complex story. A long time ago in a place far, far away. So bread wheat originated about 8 to 10,000 years ago. And Bikram, you probably know the story or know the background better than anybody. But it goes back to the Middle East.
Yes.
What happened?
So actually, there are two parts to the origin of wheat. One part is when it arose as a species. So as a species in nature produced bread wheat around 500,000 years ago. So two grasses, they happened to have fun, they hybridized, and that produced bread wheat. And then the second part of the origin of bread wheat is the humans domesticated bread wheat 10,000 years ago. And there are three types of wheat. One is called monococcum wheat, and that was the first. That's the first crop domesticated by humans 12,000 years ago. Among all crops, wheat is the first one to be domesticated.
And is that the one we call Einkorn?
Einkorn wheat is a single kernel in the spike. And then the second part is called dicoccum wheat. It has two kernels in a spikelet. And that's domesticated around 11,000 years ago in probably Syria. And then the third wheat, actually a more recent origin, the bread wheat that we actually grow, which is called common wheat. And that actually originated in Iran, maybe around 8 or 9,000 years ago in some farmer's field. Some goat grass hybridized with Emmer wheat, which originated in Syria, and that farmer was smart enough, he selected that plant, and that's our bread wheat. So it goes to some farmer. So if you're a farmer selected bread wheat, which is like world's most important crop plant.
The original selection process.
Yes,
a long time ago.
That's nature did that.
And as you said, it is the world's most important staple crop. I believe it's still correct that about 1 out of every 5 calories consumed every day comes from bread wheat.
Yes, so 1 out of 5 calories come from bread wheat, and then it's the most widely grown crop plant. So it's grown more acres than any other crop plant.
So the origin of all these species of grass, the Fertile Crescent area, I believe is what it's referred to,
yes.
Just a diversity of crops and grasses in that area?
Yeah, so the all these grasses, they originated there, they grow there as wild. And then there are crops growing there. You see wild wheats growing at the edge of the field. I've gone there. So actually, after wheat originated, these wild wheats kept cross-hybridizing with domesticated wheat and produced, essentially there were no breeders, but nature was doing breeding for the first 10,000 years. Mm-hmm. So they were cross-pollinating and farmers were selecting new types out of that. So that's how a lot of these land races, including Turkey, originated that way.
Yeah. So you're referring to for turkey red, which is not named because of the bird, as I thought for many years. It's where they found it, right? Or
yes.
where it originated from.
Yeah, Turkey probably.
Yeah. So John, let's talk a little bit. You are the curator of the current collection of the Wheat Genetics Resource Center, which we'll talk a little bit about. But in terms of science, and Bikram, you can chime in. We talk about wheat as a hexaploid, which all I know is it makes it very complex. Hexa meaning 6, has 6 sets of 7 chromosomes. So how do Einkorn and Emmer wheat compare to that? How many chromosomes do they have?
This is a great story, and I love to tell it because it happened in First World War. So the taxonomists in First World War, they are described based on looking at the wheat, 3 types of wheat. One was monococcum single-seeded wheat. Second was dicoccum, and third was common wheat, which has multiple seeds per spice.
Mm-hmm.
So in Japan, there was an experiment station director. He requested these wheats. He wrote to St. Petersburg, where Nikolai Vavilov was, his major professor. He wrote to him. He sent those wheats during First World War. They went by ship. And this student, young graduate student, he counted the chromosome numbers of wheats. That he found out that Einkorn had 14 chromosomes. They discovered that wheat had these different types of wheat had actually different chromosome numbers. So that's like the start of wheat genetics.
Yeah. So we talk about an AABB and DD chromosomes. So where do those come from?
So Einkorn is AA, and Kehara, that professor that worked, they called it, and then they hybridized these wheats also, and based on what they saw, they said the wheat, Emmer wheat, has a second genome they called it BB, and then they hybridized Emmer with bread wheat, and based on that analysis, they said, well, bread wheat has a third genome. And actually, that story is that the third genome was identified during the Second World War.
Okay.
That drama happened. So the independent Kehara in Japan and Ernie Sears at the University of Missouri, they discovered that goat grass, Aegilops tauschii, is the third genome donor.
Yeah.
So, and the Ernie Sears called it C because, ABC. But Kehara had given C symbol to some other wild wheat. He called it D. And then Americans accepted Japanese. And that's why.
So that's why.
That's why it's called AABD. But originally, Ernie Sears called ABC.
I see. I see.
His paper says ABC.
And you did say goat grass, Aegilops tauschii, which as farmers listening would think, well, goat grass is a weed. Cylindrical goat grass, is that what we call it? Yeah. But they're related, right?
That is CCDD. They share a genome in common,
Yeah, so it is essentially one of the parents.
There are many D genome species hybrids.
Yeah, okay. And that hexaploid, that repetition, there's repetition of the genomes, correct?
Yeah, so it's essentially three different genomes coexisting in one. It's like three married people living in one house. Each can be independent. They can have their own house. So that causes a problem. How do you sleep in one room?
Right.
Without messing it up?
Yeah.
So wheat has a very elegant genetic system. It won't allow, like your spouse cannot sleep with the other guy's spouse. It knows. It knows that keeps them separate.
Wow.
It's amazing.
Marital rules of red wheat. I did
ethics of red wheat.
Yeah,
it's very ethical.
Yeah, I did not know that's where this was going to go. It's a good direction.
Yeah.
Well, so it's a complex marriage. Hexaploids. There are other hexaploid species that people might not think about. I believe strawberries or potatoes. Yeah. And what is the challenge then when you're doing research on a hexaploid species?
So first of all, most of our crop plants are polyploid because just like I said, diploid are only one seed, and making polyploid almost triple the yield because there is a lot of genetic redundancy. So it's very complex to analyze it genetically. So it's a very so some like potatoes are totally messed up because they don't have this system which allows like A genome chromosome can pair with B genome in potatoes. That's why they are sterile. They're propagated by tubers. Wheat doesn't allow A genome with B. So wheat is actually pairs like a regular diploid plant, like a regular plant. Okay. So that's the amazing thing about wheat.
Okay.
It has a very, very regulated genetic system. So it's a polyploid, but genetically it behaves like a wheat. So we call it a diploid-like polyploid.
Okay, great. Well, so a while ago, you had the vision of, all these species of wild grasses that, for the most part, still grow in the Middle Eastern part of the world, that we should have a collection of these. And that was kind of the birth of the wheat, as it used to be called, the Wheat Genomics and Genetics Resource Center, 2Gs, but now we call it the Wheat Genetics Resource Center. Tell us about your vision for that and how it got started.
So my vision was when I came to Kansas State, it's a small place, essentially in the Great Plains, and not very many people have heard about this, but it's one of the greatest wheat growing belts. So my vision was when I was a postdoc fellow, I did a postdoc with Ernie Sears. He developed these genetic tools of trade. You need those genetic tools of trade, they're called genetic stocks, to map genes and to do all kinds of chromosome manipulation, do breeding. So I learned about those as a postdoc fellow. I did a second postdoc fellowship at the University of California, Riverside, and there I saw a collection of wild wheats. I was blown away by looking at the genetic diversity. And this was collected by Leonard Johnson, who was interested in the evolution of wheat. And so I had the vision that if we can develop a resource which everybody would want, and there is so much expertise in the Great Plains. Like when I came here, I went to Stillwater, I met those scientists, I went to Nebraska, I went to South Dakota, Colorado. I went to every department. There was so much expertise for diseases, insects, physiology. So we made a team, and we got this collection, and then we wrote a grant to Kansas Wheat Commission, $10,000.
Mm-hmm.
And I remember our dean, we flew in a plane to Hutchinson on a small plane. University had a plane.
Where the Wheat Commission was at that point.
Yes, the meeting. And they had never funded K-State. Until then, they were used to fund A's.
Mm-hmm.
And then I presented that this is my vision, we want to establish this Wheat Genetic Resource Center, because that's where we're going to, next yield pump is going to come from this, because that genetic diversity, nobody knew about wild wheats in the Great Plains at that time. That I know that this is where the next big revolution is. I think they asked Joe Martin, they said, well, Beck Gill's proposal looks good, fund it. So Joe Martin, I think, was my lobbyist or whatever.
Joe Martin with the wheat breeder at Hayes. And so I think this was back in 1984?
It was around 1981.
81,
yes.
Okay. And that collection still exists today. It was in...
So that was transferred from Riverside. I hired a postdoc. I went to the Dean of Agriculture. I said, I need money. We want to get this. So the best way to get their collection is to hire their person. So we hired Hari Sharma, who was finishing PhD there, and he brought this. I told him, bring that collection with you and bring that book where all this information is there. Right, John? Right? We have a copy of that book.
So he originally brought 500 different lines, mostly triticum species, but Aegilops, tauschii, and a few others. And that's where we started.
But you had to...
I have the original book of all the original collections.
Wow.
So they weren't all made by Johnson. The collections had come from like the Japanese had done a lot of collecting back in the 50s. And he had actually collections from all over, from Israel, from the Vavilov Institute in Russia. They actually started the collection process. And so we started with 500 back then, and every year we just gleaned a little bit more and more from them.
But you did take expeditions, did you not, to the Middle East to do your own collecting at some point?
We went on one. We had a Borlaug fellow from Azerbaijan, and actually our collections from that area were pretty sparse anyway.
Mm-hmm.
So as part of our entertaining him here, we were invited to go to his country and allowed to do some collecting there, I should say, because he took us to parts of Azerbaijan that the normal person really can't get into.
Mm-hmm.
I'm not sure if you're familiar, but Azerbaijan has been separated by Armenia into two autonomous states. And we also did some collecting in Georgia. And so it was very fortunate to see these things actually growing in their wild state. And Bikram is right when he said you can see a wheat field there, and there is just tons of Aegilops tauschii growing right next to it.
Yeah.
So it's not really hard to imagine how this happened 10,000 years ago.
Yeah,
because the crops are so in close proximity to each other.
Of course, right now I can go to Kansas and see goat grass growing right next to wheat, but it's a little bit, it's a different thing.
The other thing was I gave this seed. I went to Jim Hatchett. He was the Hessian fly expert, USD expert. I gave him some lines, 50 lines of tauschii, And he found like most of them were resistant to Hessian fly. That was 1980. So right away we knew that there's a lot of genetic diversity, and after that we tested for leaf rust, and it was like a gold mine. And I remember there was a headline, goat grass helps farmers, and farmers were calling up, what do you mean? How does goat grass help farmers?
Yeah, it certainly has. It's been kind of, like you said, a gold mine. You go treasure hunting in this collection that you've created here, and most of the traits, a lot of the traits that we have for stripe rust, leaf rust, viral traits, a lot of them come from that Aegilops tauschii, don't they?
Yeah. Everybody in the world is now doing wild species. I think John knows more. Everybody's working on them all over.
Yeah, it's the British have a big project going on. Saudi Arabia has a big project going on.
Everybody.
Yeah.
It's especially with Aegilops tauschii. The first reason why it's really easy to cross with bred wheat and get a nice hybrid in a couple of generations, because the sets of chromosomes between common wheat and tauschii haven't really changed that much.
Yeah.
So it's very easy to cross. Actually, virus has been the one thing that's eluded us in tauschii crosses. You can almost go and find any other type of resistance in that thing, except for virus resistance. We've had to go to more distant ancestors. However, Alan has found some pretty good wheat streak mosaic virus resistance in one tauschii line that I sent him years ago. So that was good news to hear.
Yeah, I've heard that. So let's talk about
the other thing. Just one more thing.
Yeah.
So there's the green revolution happened that doubled the yield.
Right.
Tauschii has sustained the green revolution last 20-30 years. So it's the biggest source of genetic diversity. It's the big thing.
Yeah.
It's getting tauschii introgression into wheat.
So Borlaug gave us semi-dwarfs, which increasingly yields, but you're saying Aegilops.
Tauschii has been the second biggest. It has sustained the Borlaug's green revolution.
Yeah. So what's the size of the collection, John, now?
So I just looked at it this morning, and I have some new collections of tauschii from that were part of a big study that they did in the John Innes Institute. So we'll have about 4,200 lines now. Not equally divided amongst all the species. Some have very limited distribution, and they're not that common.
Yeah.
Like one species is only found in the Sinai Peninsula of Egypt and a little bit into Israel. But things like Aegilops tauschii, that can grow anywhere from Turkey all the way in the old Silk Road countries through the former Russian states into Western China. So that probably has the biggest distribution of all the wild species.
And that tells you it's a genetic powerhouse because it can adapt all over. So it's adapted to all kinds of habitats.
And that plant, we have seen that plant growing in bogs, and we have collected it in Azerbaijan, growing right at the edge of a salt lake in a bunch of rocks that were hotter. It was like a stone massage. I couldn't bend that. Poor little plant was about 5 inches high with seed heads all over.
Survivor.
It's pretty amazing.
The collection, you have the same species essentially from different geographic locations, right?
Correct.
You treat those differently because they behave differently?
So maybe I can elaborate on this. So the Aegilops tauschii accessions, different strain, they're growing all over. So in this area, maybe there's a powdery mildew pressure. So this population will develop resistance to powdery mildew, tauschii population, but this population doesn't.
Right.
So to be able to have a chance to have some kind of every trait that you need a big collection from different areas where different type of disease pressure is, or insect pressure, or abiotic stress, or heat, or drought. So I think how many we have?
But
700?
Even with genotyping, we found that as much as 7 to 10 kilometers can change a population into different groups.
Just different environment. Yeah.
So when you have something that spans like from Turkey to China, that's a big number of different habitats they can grow in.
Which means more genetic variation.
Yeah.
We talk about being able to cross these species, and people might be thinking, well, if you're crossing two different species, I don't want to get too deep into the GMO subject, but bringing DNA from a different species in. These grasses are essentially related, though. I mean, it's so they do readily cross. I mean, like physically cross.
So we are just doing what happens in nature.
Okay.
So it happened in nature. That's how bread wheat arose. So we are essentially repeating the same process, except we're doing it in the lab.
Yeah.
Even though Aegilops has a different species name, a while ago they wanted to merge them all into the triticum genus, so they would have all been triticum anyway.
Okay. We lost a good friend from K-State here a few months ago named Bern Freebie, and Bern's job was to, some of these don't cross readily. Sometimes you've got to dig in a lot deeper.
Oh, embryo rescue. Test tube baby.
Yeah.
Test tube baby we have to do.
Yeah. Tell us what cytogenetics is. Simple.
So the genetic material, physically you can see the genetic material in a cell as rod-shaped things called chromosomes.
Mm-hmm.
So genetics is genes like, red color or brown color, that's a gene. So to map the genes on the chromosome is called cytogenetics. So cytology means seeing something under the microscope. Genetics means putting genetic traits on the...
Okay.
So Bern Freebie was the world's expert in cytogenetics. It's a very specialized area. Not too many people like to work on the microscope because they say it hurts their eyes.
Mm-hmm.
So only few people, there are few and far in between cytogeneticists.
Yeah, this is a very specialized microscope, and you can kind of dye the chromosomes. But just for example, some of these traits are hard to move over from the wild species into our modern wheat lines. I think barley yellow dwarf was one that Bern worked on.
Yeah, so wheat has a genetic system that, just like I said, it doesn't allow different genomes to pair with each other, although they're in the same A, B, and D. They're not allowed to pair with each other. Similarly, it doesn't allow any chromosome from other wild grasses to pair with wheat. So to make them pair, we have to do very specialized sort of procedures. So that's where cytogenetics, you have to be able to color code the chromosomes, and then you have to make them attached to each other, and so all that manipulation you have to see under the microscopes, and that's called chromosome engineering.
Very, very specific skill.
Very specific skill.
And we're lucky because wheat has pretty large chromosomes. So really, you don't need that much magnification.
Yeah. Large in physical size.
Yeah.
You're talking about. Yeah. Okay. Let's talk about the wheat genome a little bit. The importance of sequencing the bread wheat genome, which finally was completed in 2017 through kind of an international consortium. Why did it take so long, number one? I think we know the answer. And what does that mean now that we have that?
Around 2000, we were trying to get all these National Science Foundation, USDA, get interested in sequencing the wheat genome, and they say, are you crazy? Because it's one of the largest, it's called the Mount Everest of plant genomes, very large. So they said, no, it'll cost $1 billion and we're not going to do it. So then we organized a workshop, I organized a workshop in 2003 in Washington, D.C. We invited all the experts. So we made a plan. And the key part of the plan was there is a scientist, Dolzo, in the Czech Republic. He can actually, using flow cytometry, he could actually separate each chromosome separately in a test tube. So we could sort out these 21 chromosomes of wheat into separate tubes, even separate arms. And that was the basis of our proposal, and it got funded, and we organized an international wheat genome sequencing consortium. So this was one of the hardest projects to do, and it took us starting in 2003 until 2018.
And chromosomes are essentially assigned to different countries.
Yeah, they were assigned to different countries, and we took the biggest load. We did 3 chromosomes. Most of the countries did 1 chromosome or a chromosome, half a chromosome.
So 16 billion DNA letters in the wheat genome, if I remember correctly.
Yeah, 18 billion, I guess now. They say, 16 billion letters. And if you type 12-font type, the type will go around the world.
Yeah.
Whereas the rice genome, you type from San Francisco, it stops at Reno.
Yeah.
It's that small.
Yeah, I think for comparison, the human genome has about 3 billion DNA letters. So we're fairly simple compared to bread wheat.
And we have more genes than human.
Yeah.
We have 180,000 gene wheat plant. Humans have 30,000 genes.
Yeah.
So figure it out.
Does that complexity make wheat more durable? I mean, it's very it's a very hard to kill plant. Lord knows we try every year to kill it in Kansas.
So this is called genetic buffering. So it's almost like, there are three soldiers, three genes. So one goes out, second one is there, second goes out, third one is there. So it's called genetic buffering.
Mm-hmm.
So it is it can tolerate extremes of environment.
Yeah.
Because of this buffering caused by redundancy of the genes. Yeah.
That redundancy, though, makes it harder to
genetically analyze. That's why we had to have the cytogenetic stocks and all this stuff. No other crops have kind of cytogenetic stocks. One of the biggest breakthrough we made was working with tauschii Indo, where we were able to chop up each chromosome. And it's very simple to map genetically because you chop up a chromosome, you get rid of this part. Whatever genes are missing are present on that missing segment.
Mm-hmm.
That was a big breakthrough in wheat genetics.
So what's your role, John, with the Wheat Genetics Resource Center? I call you kind of the curator, but tell us what your responsibility is.
I call myself the curator, too.
Good. Good.
John was the best thing that happened to me.
So I used to do research many years ago, but the collection's become way too large now. It requires a lot amount of work. These wild species are not these. While they're easy to grow, they require a lot of time to clean and maintain, and some are, of course, more fruitful than others. And so my biggest job is just maintaining the collection so that we have enough seed on hand to provide to researchers so that they can do their work. For instance, Shahida Nori in the plant pathology department is working on wheat streak mosaic virus. She requires a lot of seed for each of her experiments. I just make sure we have enough stock on hand. In addition to all the wild species that we maintain, as Bikram has talked about, all these other genetic stocks that we have gleaned from not only our research, but wheat researchers all over the world. Nobody wants to maintain these because they're too much trouble. A lot of them need to have the chromosomes looked at cytologically just to make sure they're intact. Mm-hmm. Not very many people have the expertise to do this, which we have here at K-State. And plus, a lot of universities are saying these are no longer needed, so we're not maintaining them anymore. Actually, between our collection and maybe in Hungary at Matanvásár Agriculture Institute there are the only place that we're really maintaining these stocks of the whole world.
And John has a very specialized training. So these grasses, if you look at them, they all look the same. So he's a taxonomist, he's a botanist. Mm-hmm. And that's big thing because he can identify all these species. Everybody learns to identify and next year they forget. But John knows. He can look at the plant. Oh, yeah, that's Aegilops biancellus.
Some of them are still confusing.
There are, taxonomists.
And we should mention that you grow these in the greenhouse. You actually grow them outdoors here in Manhattan occasionally. But some of these ancient relatives are not readily threshable either. Or you have to separate the gloom by hand.
Yeah. When we plant them, we don't. When we preserve them, we don't. We keep them in their plant parts. It would just take too much trouble some of them are like iron. Mm-hmm. I don't even know how they probably just sit in the soil until the rain and the weather wears them down enough so they can germinate. But for preserving them, they're kept in their original plant parts.
We should backtrack a little bit to this. It is part of the domestication of wheat. Men had to select for something that wouldn't shatter and something that was threshable. So did Einkorn wheat fit that definition?
Einkorn it doesn't shatter, mm-hmm. And some are pretty readily threshable. Some are not. Einkorn, the cultivated Einkorn, which is Triticum monococcum, mm-hmm. that's grown as a crop in Europe a lot. That one's pretty readily threshable. But what we call the more wild einkorns, they still have tough glooms and they still shatter.
Mm-hmm.
So in amongst even the einkorns themselves, there's a big variation.
And Aegilops tauschii shatters pretty readily too. You can see that here.
It shatters, but it's got really, really tough glooms.
Really tough gloom.
Yeah, this gene that makes the plant not shatter is called Q gene. Essentially, this single gene turned a wild plant into crop plant.
Mm-hmm.
It made the plant shorter, made seeds bigger, head not thrash. So that we cloned actually that gene. It took us a lot of effort. And now knowing that you know the genetic code of that Q gene, you can actually, any wild plant, you can actually domesticate it by changing that gene.
Is it safe to say that the domestication of bread wheat really changed humans? I mean, into agriculture, into farmers?
Yes. Man and his bread.
Mm-hmm.
So if man is important, the bread should be equally important. That's what I will always tell funding agencies. You need to give us money, same amount of money as you give the man, because we're going to feed him bread. So I think the domestication, the cultivation of bread led to founding of human civilization, which happened in the Middle East first. When I grew up in India, we had essentially the same agriculture system that went from the Middle East to India. We were still, oxen-driven cart and, thrashing wheat when it's windy. And I mean, I lived in that agriculture. So that was a big head start. Founding of civilization, domestication of wheat is kind of all linked. And culture, everything is linked.
Yeah. So it's always been a challenge to fund the Wheat Genetics Resource Center, and it's something that the Kansas Wheat Commission has strongly believed in continuing. And most recently, it was designated as a industry university cooperative research center through the National Science Foundation, which means it's getting or had been receiving some funds from the National Science Foundation, but more importantly, brought more partners to the table in terms of private sector and university. So tell us about that National Science Foundation Center and how it currently operates.
So we were, several things happened. First of all, the seed storage facility we had in Throckmorton Hall was a small room. So we had outgrown our collections. Number two, funding was very bad. Actually, 2012, I was visiting Summit because they developed some high-yielding wheat and it has used goat grass and they wanted me to visit. And at that morning at nighttime, CNN said, U.S. Congress has cut all special projects.
Earmarks.
And of course, Wheat Genetic Resource Center was funded by earmark. And I said, oh my God. Here I am looking at, this wheat variety that was the bred with goat grass was called Norman, named after Norman Borlaug. And that night I get news. So then I even wrote an editorial in Kansas City Star. So then we, working with our associate director, he retired, founder of HPI.
Forrest
Chumley. So we had a working force, and then that's when we thought about this NSF, IUCRC, and Wheat Innovation Center, and New Center, and the whole thing. And that's where it started.
Yeah,
that we needed new facilities, we needed new funding, and luckily this was one of the first IUCRC and crop plants. Otherwise, until then, they were all in Silicon Valley.
Right.
Computers and engineering and others.
Yeah.
So that has been a big, and the founding of Kansas Wheat Innovation Center and the new gene bank here, and the lab, it has been just amazing. People get amazed when they come visit here.
There's several research projects going on under this umbrella, John, and so give us a brief rundown of what some of that work is doing.
Well, we're really working hard on sawfly with a lab at Colorado State University. That's been a big project for the last couple of years. Recently, a new project that was funded was looking at root morphologies for drought resistance. Evidently, they're learning that some of the roots are sharper than others, and plants that have these sharp roots are able to penetrate droughty soil better than others. So there's two labs that are working on root morphology, one at South Dakota State and one at Texas A&M, and they actually have a really interesting growing the wheat plants in these tubes and running MRIs on them so there it can actually look at the root structure instead of digging a plant up out of the ground or growing it in a small pot, which is not naturalistic. They grow in these huge long tubes that are 6-7 feet long, so they can look at the intact root structure.
Fascinating.
So that's pretty interesting.
Do you think we've even scraped the surface, Bikram, of the potential in some of these ancient grasses? Still a lot more to be discovered, in your opinion?
Oh, yeah, I think so. There's a lot more to be discovered. We were pretty much 1 or 2 labs, were working on wild weeds for many, many years. Everybody was asking us for the germplasm. But even now, I think they're doing a lot of genomics. I'm not even sure if they're still doing any experimental work, where you make hybrids. I doubt even if still.
Actually, we were kind of. There are probably at least 15 species that have not even been used at all. So there of these what we call wild weeds, which are shattering, tough gloom plants. Basically, there's almost 21 species in the genus Aegilops, and there's another 15 in the genus Triticum. So there's probably about 15 of those that haven't even been used at all. We are lucky. Alan Fritz, the wheat breeder, loves to work with these lines. He started with Aegilops tauschii. He went to another species of Aegilops. Then he went to another species of Aegilops. And he has recently asked me for seed of 4 more species. So Alan is good at making these crosses and interested in these wild species.
Actually, when we look at what kind of material from wild species has made the most impact, it's actually the breeders working with wild species. We had some material from Oklahoma where a breeder produced this material. Similarly, we had some material from South Dakota, breeders that. So what we did was, using our advanced cytogenetic tools, we defined the genetic variation. They were able to transfer the small amount of genetic material and for traits for Hessian fly resistance and wheat streak mosaic virus. That original material was actually produced by breeders who made their crosses in the 1920s, but they couldn't figure it out genetically. And we figured it out and we refined it, and then now it's used in production agriculture. So.
Yeah. We've mentioned some of these traits of virus resistance, rust resistance. We haven't mentioned that some of these grasses are super high in protein too. I think Alan is working on a little bit of that. If we can just get a little of that protein into our modern wheat lines.
Yeah, some of the Emmers have 17% protein, 20% protein.
Yeah, Mary Gutierrez,
that USDA.
The USDA scientist, has done a lot of crossing with a lot of our wild Emmer species and has found some really high protein lines.
Yeah. I should mention this collection is publicly accessible, correct? I mean, you get requests from all around the world.
All around the world, almost weekly. I usually send out about 100 different requests every year. Can vary from a few lines to several hundred lines. It depends on what the researcher's working on.
A couple of years ago, last year. I mean, you won't believe it. Somebody from Dubai asked for genetic stocks of wheat, monococcums. Scientists. So, I mean, all kinds of people are asked from Dubai. I was blown away. I said, Dubai, some scientist there wants our stocks, so I forwarded to John.
And just last week, I got a plea. I'm not sure how to handle this yet, but I got a plea from a Ukrainian scientist that wheat streak is becoming a problem over there. And I'm not sure how I'm going to get the seed to them. Sometimes it's not easy to get seed to people that request it.
Yeah.
You have to find a way to send
seed. I know. They might not get Federal Express. I don't even they have mail over there.
Shipping seeds a little bit tricky. What you gentlemen have seen in terms of change in technology the last 40 years, and I'm talking about just like sequencing technology that wasn't available to a guy like Norman Borlaug. I mean, how far has that come, and what do you think is possible in the next decade?
I think the sequence has totally changed the game. So now it's not a problem analyzing, what kind of genetic transfer is there. It's the hard part of the growing plants, making crosses, producing material so that you can analyze it. So, so the sequencing has totally changed the game changer. I mean, I have a vision where totally give my new vision of WGRC. We are maintaining single species each accession. Now I have a vision. You can actually collect everything, grow everything. Just type it by sequencing, and then you know what accession it is. You can actually identify each accession now, but it's based on sequencing.
Mm-hmm.
So you might as well grow everything as a big population.
A lot of work.
A lot of work.
A lot of work.
If you want to test it for something, salt tolerant, just grow the whole population somewhere in the field, and whatever survives, you can identify sequencing what it is.
Yeah.
What strain?
Any opinion on gene editing? Is it a tool that with combined with the sequencing knowledge we have that can make large leaps?
So what sequencing has done is it has identified all the gene candidates, all the genes in the wheat plant. So that was essential. If you don't have the gene sequence, the sequence of that for whichever trait it's controlling, you can't do anything. But now what CRISPR allows is to make that changes in the gene, then you can make a new trait. So I can give you example. Let's say you have a wonderful wheat, but its leaf rust gene got defeated. It became susceptible.
Mm-hmm.
Well, CRISPR will allow you to fix that gene, and you can replant that wheat, make it resistant. So now that we know all the genes are known, combined with CRISPR technology, we'll be able to do a lot of kind of novel manipulations, very precisely change the gene, tinker it, and then, get a new germplasm. So it's a big
Sky's the limit there. Let's talk about these collections. There's probably only a handful of these around the world. One that we're very proud of that's here in Manhattan, Kansas, in a building built by farmers. But this is like preserving a language, and you've got to have some security in knowing that you'll be able to keep this collection. Is there redundancy among these collections, and is there a big backup? I believe there's maybe kind of a doomsday seed vault somewhere. How are these seeds backed up?
Yeah, I don't really like that term doomsday.
Worst case scenario.
But so they're around the world. We're a small gene bank. The large one in the United States is in Fort Collins, operated by University of Colorado, with a satellite for small grains at Aberdeen, Idaho. So for wheat, that's the major one in the United States. And they fill lots of seed requests. For rice, it would be in the Philippines. So different crops have large gene banks in their center of the most origin. But for wheat, there are maybe 5 major places where these species are stored. And one would be in Kyoto, Japan. The other one is in Gattersleben, Germany. Hungary, as I mentioned, Morocco. Actually, until a few years ago, CIMMYT used to keep a lot of wild species, but I don't think they do it anymore.
CIMMYT being a wheat research in Mexico, founded
Yes.
by Norman Borlaug.
Right.
Let me just add to John. There was a big seed bank in Akarda in Syria. So when the Civil War happened, that gene bank got taken over by rebels. And then they had to move the seed back to Morocco. And then John supplied a lot of accessions because they're duplicated here to their gene bank in Morocco. And the other thing is that seed bank they took over in Syria, these are some really criminals because they have not destroyed the collection, they're trying to sell it.
Oh, wow.
They're walking, somebody was paying them $1 million, they said that's not enough.
Wow.
Okay, so the other part of the story was about 15 years ago, I guess. These major seed banks of all crop species decided that they would have this doomsday bank, as you called it. And it is for really long-term storage. Things are put in there and they're just kept there for eternity, we hope. And that is in Svalbard, Norway. And it's built in the permafrost there. It's only entered a couple times a year. Most people put the seed in there and it stays there forever. The situation with wheat was really unique. The seed bank at Aleppo, Syria was compromised by these rebels. The seed bank was moved to Morocco, and Morocco actually had to petition to go into the Svalbard gene bank and bring samples of barley and actually a few other crops, not just wheat related, and grow it in Morocco because it was gone. There was no other place to get it in any other gene bank. But we did have some of those too. So I did supply them with seed. So there is this seed bank in Norway, but it's basically the stuff is put in there and hopefully not going to be needed unless it's a real disaster.
Let me pitch in here about doomsday seed bank. So this genetic diversity that we have in the wild wheats, it arose in nature over the last 500,000 years. I said wheat originated 500,000 years. So it evolved in that time. So it's like a money in the bank, and we are now cashing in.
Mm-hmm.
The problem with the gene banks is, long-term problem is, it's not evolving anymore. It's not exposed to, it's not growing in nature.
Yeah.
So this is what keeps me up at night. What we have is, in a way, it's a doomsday bank because it's useful for the short term, but it's not useful 100,000 years from now. We have to, and those species in nature are disappearing, those centers, the original crop plants. So what are we going to do if we need a new genetic diversity 100,000 million years from now if those species are not growing in nature?
Yeah.
So this is a big question.
Yeah. Right now, wheat growing out there is adapting to changing climate.
Yeah. So,
but that seed is not.
That seed is not. So
Yeah.
this is a big issue, and that's what I'm thinking these days, what we should do next.
Yeah.
Because as far as we know, no one's, like a lot of these were collected in the 50s, 60s, and 80s, and no one's gone back to those exact same locations, collected plants from those same locations. Okay, have these changed in the 30, 40, 50 years since they were originally collected?
So Bikram, I know you are from India, and you talked about your farming history there. You're as retired now, you are doing some work in India. Tell us about that.
So actually, after I retired, I still get invited. I have a lot of pedigree. My academic grandsons, my students, they are working in all these countries. So I get invited. So it's what I'm doing is more sort of academic work. I go give seminars and talk to students. But also I've been working in Punjab, where I come from. It's one of the biggest wheat growing area, just like Kansas in the U.S. So they have a lot of issues with, so they were developing their new agriculture policy. So I was able to interact with their government agriculture department. Recently, Morocco also is doing the same thing, developing their new agriculture policy. And again, for genetic resources, they asked me. So I'm doing a lot of this. I've gone to Turkey. A lot of these are all, again, interested in genetic resources. How can we use those in our crop improvement programs? So that's about essentially the extension work for WGRC.
Bikram and John, thanks for joining me here on the podcast. My thanks to Bikram Gill and John Raupp for joining me on this episode of the podcast. Remember, if you have a question about this podcast or you have a topic you'd like us to cover in the future, please email us at kswheat@kswheat.com. I'm Aaron Harries. Thanks for listening.